start. I would like to give a short overview of the MBST technique. And to start with, currently MBST, which is a magnetic resonant therapy, is used in all types of musculoskeletal diseases. It's used from osteoarthritis to osteoporosis, sports injuries, and post-surgical. The company was started 25 years ago. We currently operate in 350 centers worldwide with 650 devices installed. In this year we will have 45,000 patients treated at the centers, which sums up to a total of around 370,000 patients treated since MBST started. We currently registered in Europe through MDR, UK, and the Emirates country, Saudi Arabia, and pending registration process in United States, China, Japan, Canada, and South Africa. And in Germany, where the technique is used for the longest period, it's already paid by private insurance companies. I want to give today a short idea on how MBST works. I want to give you a short overview why it works in the cells and what the clinical studies reveal. I will mainly try to give some clinical examples and sum up on how we see the future use of MBST therapy. So the MBST started with a finding, and the finding was that serial MRI scans lead to an improved outcome of patients. So patients of serial MRI scans reported increased activities, increased mobility scores, and these results were initially seen as placebo effect by the machines. But when further the effect of energy transfer by resonance was studied, a research group in Jülich, Germany, started to more deeply discover the effect of an MRI resonance on cells. And a short recap what resonance means. Resonance is an energy transfer between two physical bodies which operate on the same frequency. An example everybody knows from medicine is skin cells. And in skin cells, if UV light is applied, UV light have the same resonance than the electrons in the cells, and it leads to a jump of the electrons, which leads to the destruction of the cell, skin burning, and cancer disease. And in the same way, you can apply a resonance in water protons. And if you use a radio frequency and apply these resonance, the energy of the proton is changed and the loading is changed. And this leads to changes within the cell metabolism. In sum up, this technique is already used in MRI, but MRI is using this technique for imaging and only doing a small excitation. So it doesn't use the full resonance effect, it only excites the protons. And what the MRI does, it aligns the magnetic field, which is called a T1. It affects the resonance of the spin, and these two parameters, T1 and T2, are switched on and switched off all the time. And as all the tissues have a different behavior in their magnetic alignment and in their resonance, you get a gray-white contrast and can see the different structures. And for this, because the effect is so subtle, you need a very strong magnetic field. What MBST does, it's using only the resonance effect, but it keeps the resonance effect for a certain period of time, that there's an efficient transfer of energies to the protons in the cell and changing the loading of the cells. So the magnetic field is only used to align, but not to have any therapeutical effects. The therapeutical effect is by the resonance in the cells. And by the resonance, it directly affecting the cell metabolism, the loading and the nutrition of cells, and this leads to quantum biology changes, which I will show later on. one very important question normally comes is, are we aware of magnetic therapy? So everybody talks about magnetic field therapy, magnetic therapy. This is something completely different, because the magnetic field therapy only use the alignment of the protons, so only the magnetic component of, so there's no resonance effect and no energy transfer to the protons within the cell. What the magnetic field is doing by switching it on and off, piezoelectrical effects, so micro changing of the loading of surfaces occur, and these micro changes on the surfaces lead to a biomechanical reaction, and this is activating capillarization and ionic transfer in the cell, nutrition, and therefore currently the technique is used successfully mainly in bone diseases with overuse and in muscles where the structure can be improved. But it's not a direct effect within the cell. And therefore, magnetic field therapy and the MBST technique using a complete different physical mechanism and also a complete different biological reaction. So now we come to the stage why it works. So after this effect of resonance was discovered, cell studies were done where it was investigated how this effect changes the cells. In the first step, they only watched the viability and the apoptosis, the cell death. Because first of all, it has to be sure that there are no side effects. But the interesting finding was that not even the MRI have no negative effect on the therapy. It has a positive effect that the apoptosis of cells, which is here induced, you see it in the green-red, and here without the green there's no apoptosis. So even an induced apoptosis can be stopped by using the MBST, and the viability, the black spots show decreased viability, are positive affected by the MBST. And in addition, as a side finding, they could reveal that the number of cells was also significantly increased in the blue group where it was treated with MBST. As these studies were done in chondrocytes and I think osteocytes, they tried to transfer the same to other cells to see if this is a sustainable effect through different cell types. Therefore, they used neuron cells, which are known to have a mixed structure of growth. There's two types of cells, the neuron cells itself and the Schwann cells which covering the neurons. And the Schwann cells are, that they the control cells of the neurons. They control the nutrition, the growth, the branching. And in neuron cells, we applied the MBST technique. This is a very significant article from a research group in Vienna. And it was seen that using MBST, there were more primary neurons. So new neurons were developed in the cultures, highly significant. The branching of the neuron was significantly increased, so they grew in a physiological way and the length was increased. And the interesting finding here was that this increase was due to an increase of tubulin beta. This is a single induction pathway in the cells, triggered by the Schwann cells, which leads to the nutrition and metabolism increase of nerve cells. So it was concluded in the article that the MBST technique has to directly affect the signal induction pathways within the cells. And therefore, in the next phase of studies, already very good explanatory models for signal induction were used to better or further investigate MBST. And one very well-known described model by rheumatologists is the osteoarthritis model in cartilage cells. You have different signal induction proteins which are responsible for the catabolism of the cells, and you have different proteins for the signal induction for the anabolism, for the growth of the cells. And they applied the MBST technique and measured each of these factors in normal and osteoarthritic affected cells. And the interesting finding here were all of the inflammatory catabolic went significant down, especially the interleukin 6 and 1, which are responsible for the early anti-inflammatory and pain reaction. But not only these effects were downgraded, in the same way the anabolic effects and the cell metabolism was increased. And this was revealed in these two articles from the Vienna research group. And they concluded out of their article that it looks like there's a direct link between the resonance changes in the loading of the spin and the signal induction pathways within the cells. The remaining question was how directly it can really affect the signal induction pathways. And for this, we studied deeper in the hypoxic signaling pathway. The hypoxic signaling pathway, which was discovered, I think, 1992, is currently seen as a basic mechanism for metabolism, cell growth, angiogenesis, anti-inflammatory, autoimmune, ischemic reaction. So all reaction of the body to damages, mechanical damages, nutrition damages, drug damages, is triggered by hypoxic signaling. The reason why it's called hypoxic signaling is that the hypoxic situation is the best described stress to the cell if you decrease the oxygenation of the cell. And all these processes were seen when the cell had no oxygenation. Then they derived different factors, which they described as hypoxic-induced factors. And all these factors were also seen in cell growth, in autophagy, in autoimmune reaction. And in, I think, 2017, the Nobel Prize for Medicine was also for the hypoxic signaling pathway. And the second thing we know about hypoxic signaling, that this is influenced by radiation, it's influenced by magnetic fields, so we know that physical parameters can affect hypoxic signaling. Also day and night time, the circadian clock. And therefore MBST was applied to the hypoxic signaling pathway. And this is the most significant finding on the cell level. This was just published this year now in Science, the bigger article. And what it revealed was that MBST, so magnetic resonance therapy, affects directly the hypoxic signaling with a significant increase, the positive hypoxic signaling, and the circadian clock, which is, let's say, the trigger within the cell. So it looked like that it directly affects the pathway of normal and less oxygenation within the cells. And to have one very concrete example what it means for the therapy approach. The hypoxic signaling is responsible for the glycolysis in the muscle. So if you have a top athlete who's running, he has a regular glycolysis where the glucose is transformed in ATP 32 per glucose, and you have high energy in the muscles. But we know if you have any type of stress, less oxygenation, less training in the muscles, the hypoxic signaling starts and it leads to lactate glycolysis. So the glucose is only split in 3-4 ATPs, and you have a high concentration of lactate in the muscle, which leads to further damages and destruction of the muscles. So therefore we studied the effect of MBST to the early hypoxic effect of the lactate genesis. And here you see the results. In black you see this is the regular level of glycolysis in the muscle cells, which was measured in vivo. Then there was hypoxic stress, and you see the glycolysis drop down to this level. In the same way, the lactate was increasing. So the muscle was damaged. By applying MBST in the same situation, even with the hypoxic stress, the glycolysis recovered to a regular level, So the hypoxic signaling could be stopped and the regular glycolysis and metabolism of the body could be activated. And even more significant, there was no production of lactate. So the negative side effect, similar to the interleukin 6. So everything where the body starts a signal induction pathway for destruction could be inhibited. And this explains why MBST can treat different type of conditions and have, on the one hand, a pain relieving early effect, anti-inflammatory, where you can increase mobility, but also having a long-term effect for regeneration of the cells by activating the regenerative process. Now coming to the clinical data.